I am Daniel Luksic, and I am a medical science liaison with Natera's Oregon Health. Today, I will be presenting a paper titled Clinical Integration of Genome Diagnostics for Congenital Abnormalities of the Kidney and Urinary Tract. This was written by pediatric nephrologists and medical geneticists from the Netherlands and published in the clinical journal of the American Society of Nephrology. This is a review article and serves as an overview of the genetics of congenital abnormalities of the kidney and urinary tract, or CAKUT. Although CAKUT is most frequently developmental defect in humans, genetic identification and classification of these disorders are hampered by the heterogeneity of their presentation, which is driven by the high phenotypic variability, multifactorial inheritance, incomplete penetrance, and variable expressivity. CAKUT is due to disturbances in molecular development, and patterns have been hypothesized, which can attribute defects in certain time and space to the development of certain phenotypes. The phenotypic complexity of CAKUT is often clinically subdivided into anatomical order, the first being parenchymal defects of the kidney, which result in agenesis, hypoplasia, dysplasia, or multicystic dysplastic kidneys, or ureteral defects, which result in ureteropelvic junction obstruction, ureterovesical junction obstruction, and vesicoureteral reflux. Lower urinary tract malformations describe bladder exstrophy, bladder agenesis, posterior urethral valve, or urethral agenesis. CAKUT is by no means limited to these anatomical boundaries, and co-occurrence of several types of defects are commonly seen together in patients. Furthermore, CAKUT is not always permanent, and some hydronephrosis and low-grade vesicoureteral reflux may resolve during development. Standardized guidelines to uniformize clinical decision-making for individuals with different CAKUT phenotypes have not been developed due to the absence of systematically performed longitudinal studies. In part, the unavailability of such studies prevents clinicians from discriminating between patients with an impaired survival of kidney function progressing to kidney failure from those who have a more benign course. Furthermore, parents or family members of a child with CAKUT often inquire about recurrence risks, and an improved understanding of the molecular etiology is needed to provide these answers. We will now review what is known of the etiology of CAKUT. The implementation of next-generation sequencing based on genetic testing has significantly enhanced our knowledge of the etiological basis of many diseases and has added to the personalized patient care. Here, the authors highlight the essential aspects of the complex etiological landscape of CAKUT, beginning with monogenic causes. These are assumed to be responsible for 10 to 15% of cases of CAKUT. This understanding is based on segregation within those with a family history, where the genetic defect is usually considered to be autosomal dominant with incomplete penetrance and high variability. CAKUT has often been seen as symptoms of genetic syndromes with multiple organ systems, where it is again presumed to have a de novo mutation. Over 50 genes have been associated with monogenic forms of CAKUT, some of which are known to have other non-CAKUT renal abnormalities. Next, we have structural variation, which concerns large portions of the DNA being missed or duplicated. Structural abnormalities, otherwise known as copy number variants, take the form of deletions or duplications of regions of DNA. Although these variations are common in healthy populations, it has been observed that larger deletions or duplications are more common in individuals with CAKUT than in healthy controls. Much like monogenic disorders, it's estimated that around 10% of cases of CAKUT are attributable to pathogenic structural variants, which are characterized by phenotypic variability and, again, reduced penetrance. Of all cases of structural abnormalities being recognized as the case of CAKUT, 65% were caused by only one of 6 common deletions. A final genetic mechanism of inheritance is polygenic mechanism, or the cumulative effect of many genetic variants which individually have a small effect. These common variants are assumed to play a role in CAKUT in the same way that they are known to contribute to risk in other disease. A polygenic risk score is the sum total effect of these multiple small effects, and in the case of other diseases can confer a risk equal to that of monogenic disorders. In order to establish the role of these common variants and generate a polygenic risk score, variants must be first discovered in genome-wide association studies. These studies require large numbers of cases, controls, and few such have been completed. Once suspected variants have been identified, they must then be confirmed in separate trials, which often fail to reproduce the significance of the original association. Despite these difficulties, it's likely that eventually there will be polygenic risk scores available for CAKUT, which will possibly provide an explanation for a significant amount of CAKUT's occurrence. Another mechanism of disease is epigenetics, or the modification of gene expression. Control of gene expression may be influenced by modification of the DNA itself or by changes to expression of microRNAs, which affect the DNA's translation and protein synthesis process. To date, studies have found a very small percentage of CAKUT cases related to epigenetic effects, though studies of these epigenetic effects are still in an early phase. Finally, the authors review the environmental effects that can influence congenital renal abnormalities. There are several environmental effects known to associate with congenital renal abnormalities in utero, such as maternal obesity, diabetes, or drug toxicity. Low birth weight, which can be taken as a proxy for in utero environment, is also known to correlate with reduced nephron function. Environmental effects in the postnatal period may also play a role in the development of CAKUT. The gut microbiome, for example, is known to be affected by and affect renal function. Next generation sequencing is becoming cheaper and more widespread and promises to increase the identification of disease-causing genes and the direct implementation in genetic diagnosis. The authors see great importance for improvement of CAKUT DNA testing by creating awareness among clinicians. A genetics-first approach could result in considerably shorter and less complicated diagnostic process and help avoid unnecessary testing and improve outcomes. They feel that standard genetic testing of syndromic and severe CAKUT cases is reasonable and should already be considered globally. The authors see whole genome sequencing as an eventual standard of care, pending our ability to interpret the data that it reveals. This exhaustive technique is able to evaluate monogenic, structural, and common variants. Standard classification of CAKUT is essential to develop the framework that will enable this interpretation. Proteomics and metabolomics may also eventually be added to provide a complete clinical picture of the patient. In summary, CAKUT is a heterogeneous, complex set of phenotypes with diverse etiologies, for which, despite increasing genetic advances, difficulties remain to predict outcomes and estimate recurrence. A personalized medicine approach still faces hurdles of data analysis and implementation, and further understanding of the molecular basis is required. Thank you very much.